All right, can you guys hear me? Okay, it does show I have audio. I won't have
Yes.
I won't have a camera, and like my audio is coming from my closed laptop. So just this is my first time using this setup, so bear with me if there's some weird stuff going on. It's going to be a learning experience today. So I'm going to share my screen. Maybe. That's not the screen I expected it to be on. Oh, might not work. I wasn't expecting it. Okay, it does show up. Okay. We'll give it just a minute. Let people filter in. Okay, I think we're probably gonna have that's as many people as we're gonna get. That's okay. I don't want to wait too long to get started, so I'm gonna go ahead and just get going with this. So we're doing a week 6 review. Um, we're going to be our agenda today talking about chapter 6, which is genetics, chapter 8, which is genetic engineering. The most missed topics from those will have maybe 2 or 3 questions for each chapter, and go over that stuff, kind of briefly glaze over like the content for those things. I feel like I spent a lot of time going over what a genome was. So I feel like we did pretty good justice. Why is that not on? That is weird. Okay, this is off from my pen. Anyways, talking about genome and genotype, we did kind of do that. But remember, a genome is the sum of the genetic material of an organism, including any extra chromosomal DNA, which might include things like plasmids, for example. Right. A phenotype is basically what express your physical expression. Phenotype, physical, genotype, your genetics, your gene, your makeup there. All of your genes are made up of DNA. All cells have DNA for their genes, and it's double-stranded DNA. Normal double helix, double-stranded DNA. Whereas viruses, they have either RNA or DNA. It's single-stranded, double-stranded. If it's single-stranded, it can be positive sense or negative sense, right? DNA or RNA. This is R for sure, RNA. And both of those can be either double-stranded, single-stranded, if it's single-stranded, positive or negative sense. DNA itself literally means deoxyribonucleic acid. RNA is ribonucleic acid. With DNA, the four bases that we talked about, adenine, A, T is thymine, C is cytosine, G is guanine. And this, again, is existing within a nucleotide. Adenine is not a nucleotide. So I want to be very clear about that. Adenine is not a nucleotide. It is a nitrogenous base that is part of a nucleotide. So what's a nucleotide? We should know this by now, but just to remind everybody, it's a sugar linked to a phosphate. And this is a 5-carbon sugar that is based on ribose. So you're either ribose or you're removed an oxygen and you are deoxyribose, right? So there's a 5-carbon sugar linked to a phosphate, and linked to that sugar is a nitrogenous base. And that is your A, T, C, or G. This whole structure is the nucleotide. And this is our repeating unit in both DNA or RNA. The difference between DNA and RNA is the sugar, you're either deoxyribose or you're ribose, and the bases. In DNA, it's ATCG. In RNA, it's a UCG. So we're replacing the T with the U. All right. So that's the gist of that there. Right. AUCG. Okay, what else do I want to say? We have the two anti-parallel strands that run in opposite directions, which refers to that sugar and phosphate basically alternating. So this sugar phosphate group here is what alternates. So in one strand, you'll have sugar phosphate, and then the base off to the side, and another sugar phosphate, and the base off to the side. But the other strand runs in the opposite direction, where that base is hydrogen bonded to the other base, and it'll still be linked to a sugar, and it'll be linked to a phosphate like this. And we drew this and went over this in our review. And if you guys are in my online class, there should be a video posted in your discussion session where you can see where I drew all of this out for the ladder and then described how this is going to relate to our replication and things like that. So this is important that we understand this is what anti-parallel means for all of that. Okay, so I'm going to erase this because this is looking sloppy. Move on. This is not the best at erasing either. All right. Moving on. Let's see. Where are we at? We had the anti-parallel strands, and the direction is in 5' to 3' in one side, and the other side is 3' to 5'. Remember, the prime is literally just referring to which carbon on the sugar is at whatever end. And I'm not going to draw the whole structure, but we number our carbons based on what they're attached to. If something's attached to a big old nitrogenous base, for example, let's pretend that's our nitrogenous base, then you might call that number 1, and number 2, and number 3, and then number 4, and number 5, based on whatever they're bound to. So if we had, a phosphate on one of them, like a phosphate, then we would number accordingly in whatever direction that we needed to go. It's not an accurate drawing, and I'm not going to test you on it. So I don't want you focusing too much on that specific drawing, please, because it's not meant to be accurate, and you're not going to get tested on it. You're not going to have to recognize it. But for reference on what 5' to 3' and 3' to 5' mean, you should have learned that if you took GOB, which is your Gen Chem, Organic, and Biochem, like mini course. I hear a lot of people calling it organic chem. Trust me when I tell you, that is not organic chemistry. It is primers just on dip your toe in basics on that stuff, okay? So we should have been introduced this in that organic and biochem side of GOB if you took GOB. All right, if you took Gen Chem, then this maybe is a little bit more new to you. but that whole concept is still the same. We number our carbons based on what they're attached to. So if this is attached to a base, and then one of them's attached to a phosphate or whatever, whichever carbon is at that end, we basically number the carbons, 3, 4, 5. And if the 5 is at one end, we call that the 5' end. If a 3 is at the other end, we call it the 3' end. And whatever is attached to the phosphates and the bases and whatever kind of in between and all of that, okay? Attached to some of these carbons or attached off to the side or whatever. I mean, that's how that is numbered. So basically based on your sugar, phosphate, sugar, phosphate, sugar, phosphate. I said I wasn't going to draw it, and then here I go, right? Just playing games with me. And then the base is in the middle. And again, they're hydrogen bonded together. Hydrogen bond to a base, hydrogen bonded to the other base. All right, so this is a nucleotide, and this is a nucleotide, and this is a nucleotide, like this is. But you can see when I draw it like this, that they are definitely oriented in opposite directions. That's what anti-parallel is. Our backbones are in the opposite direction, right? And these two sides of our double helix, remember, this is one strand, and this is our other strand, and the two sides of our double-stranded DNA are held together in the middle by hydrogen bonds. Those are weak, they're just attractions. And the way that replication works basically relies on the fact that these are just attractions and it is weak, right? And so whenever we separate those, we're literally just pulling apart the hydrogen bonds, okay? All right. Okay, so that's how replication will work. I'm not going to walk through all of replication because, again, I walked through that in our classes. And again, if you're online, don't think you've missed out. I posted a video of how we did it in the classes. So if you need another version of that walkthrough, if watching the lecture wasn't enough, you can watch our in-class video recording as well. It's posted in your discussion area on Canvas. But the gist of it is that we have our DNA ladder that gets opened up, and we have one side of the DNA and the other side of the DNA. And if this was 5 to 3, and this is 3 and 5, remember, we only move in 5 to 3' direction. So as we're making our new strand, copying that one, let me make a new color. As our new strand, it can go in continuously 5 to 3. This other side cannot go in continuously this way. It can't do that because DNA polymerase only adds in the 5' to 3' direction. So it can only add in the 5 to the 3. So as this replication fork opens up, we can go in continuously on one strand and discontinuously or in fragments on the other strand. Those fragments on that other strand are called Okazaki fragments, right? And the Okazaki fragments on that other strand are linked or sealed back together by DNA ligase, right? Again, Okazaki fragments, and that other strand that's made discontinuously is the lagging strand. Leading strand is continuous. Lagging strand is made in Okazaki fragments, and we seal those fragments together using DNA ligase. All right. So again, that's the summary. Oh no, that's not an eraser. That's the summary. And if you want to see me walk through it in a little more detail, please watch the recording if you weren't able to watch it during class. All right. Next is RNA. So this is another one that I want to be sure that we are clear on before we get towards the exam. And that is replication is DNA being copied into DNA, period. All right. Transcription is using DNA as a template to make RNA. Anytime we're making any kind of RNA, If we have to use DNA as a template, period. If you're making tRNA, transfer RNA, you had to use DNA as a template. If you're making ribosomal RNA, rRNA, which is what makes up the majority of your ribosomes, that's what they're made out of, those were coded for in the DNA. Obviously, the most common thing that we talk about with transcription is mRNA, messenger RNA, which is a working copy of a gene. And then we're using DNA as the template for that as well. But all RNA was synthesized from a DNA blueprint. And we do transcription DNA to RNA. We're making a working copy of whatever that DNA was. Period. Whatever kind of RNA you're making, we're making a working copy of the DNA. And we use the analogy of the cookbook, right? That big giant cookbook of anything you could ever want to make as a recipe. And it's so big, you can't carry it around. So you leave it in the pantry, which is the same thing as the nucleus. But you'll just copy down a recipe for spaghetti and meatballs. You copied down that recipe for spaghetti and meatballs. It's still written in English or whatever language you speak, right? Still written down in the same language. It's just handwritten temporary working copy. That's the exact same thing here, all right? That's what transcription is. Like a scribe writes something down, transcription, like a scribe, script, okay? All right, the next part is translation. And that is using that RNA working copy to make protein. That's not the best I've ever written protein, but hey, you know what I mean, okay? So we're making protein out of that. It's really bad. It's really... So we're not using RNA to make amino acids, not specifically. We're using RNA to build protein using amino acids as the language. So this is where I want to make that comparison. When we were doing replication, we were building DNA using nucleotides, right? When we went from DNA to RNA, we were building RNA using nucleotides, okay? In the case of translation, we're going from RNA and reading the RNA codons, which are 3 nucleotide set. AUG codes for an amino acid. CCG codes for an amino acid. GGA codes for an amino acid, right? A specific amino acid. And those amino acids are linked together by peptide bonds inside of the ribosome. And then that is going to build our protein. So again, RNA isn't coding for amino acids, really. It's coding for individual amino acids that are going to build a protein. That is the more important thing that we're focusing on. So RNA codes for, yes, individual amino acids, but they're being linked together by peptide bonds to build proteins. So that's really what's going on there, okay? So when we make RNA through transcription, that process works very similarly to replication. It's just instead of building 2 strands of DNA, we're building 1 strand of RNA. But other than that, it's pretty similar. When we go through transcription, RNA polymerase is the enzyme that makes the RNA. It reads the DNA and then adds on the appropriate nucleotide. A binds with T and T binds with G, or sorry, excuse me, A binds with T and C binds with G in our DNA. But in our RNA, if there's a T, we'll put an A, but if there's an A, we'll put a U if we're building our RNA. So that's going to be the main difference there. And again, I go over this in more detail in the other video. This is me summarizing this here. So, right. So anytime that we're making any RNA, that'll work the exact same way, but we always start at a specific spot that I feel like is getting left behind a little bit on some of our discussions here. All of transcription, RNA polymerase starts, okay, I meant to put polymerase. RNA polymerase starts at the promoter. And this is important for understanding our operons. On DNA, the promoter is a thing on DNA. So imagine you have a DNA strand. Here's your DNA ladder, whatever. And on that DNA strand, there's a specific area called a promoter. And then this is where RNA polymerase will bind onto and open up that strand and then copy one of the strands to make our single-stranded RNA. But other than that, it works this very similar. How we're opening the strand, how we're moving along and elongating, otherwise works very similarly to replication. There's three major steps of transcription, which is basically our initiation, which is binding to our RNA polymerase, our site, which is that promoter. Initiation is binding to the promoter. Elongation, adding the nucleotides. And this literally is where we're talking about if your DNA has A, T, G, then your RNA will copy over U, A, C. So that's the elongation and how we're reading it and what we're going to be adding on for nucleotides as we're building our new RNA molecule. And then we get to a termination sequence. And in transcription, it's just a termination sequence. That's all you need to know about it, okay? All right. I know the book goes into more detail, but this is all we need to know for my class. All right. There's a major difference between bacterial genes and eukaryotic genes. And this is one that got a little bit left out of my lecture or in our in-class review. It's in the lecture. I'm sorry. It's in the lecture. It's just we didn't go over it much in our in-class session because I can't cover everything in the in-class session. So one of the things that got left out in some of the classes were introns and exons. Eukaryotes have these. Bacteria do not. And so the introns are basically waste. We don't need them. And so you'll have your RNA copied from your DNA, and it'll have code that we need, and then these long areas where we don't need, and then code that we need, and then a long area that we don't need, and etc. If you need it, I'll label them here so we don't get confused. Don't need and then need. The ones that we need, we call those exons. The ones that we don't need, we call those introns. And these will be in the DNA, and then we copy them over into the RNA. And so what eukaryotes do with this is they literally splice out the introns and join together the exons. And so what they'll do is they'll fold up that region and join those two ends together so it would look like this. You get the idea, right? And then we will splice those out. It's literally called splicing. And our cells do that for us to join together our coding regions on our mRNA. So this will be our working mRNA after we have spliced it out. And only eukaryotes have that. Only eukaryotes have introns and exons. Bacteria and archaea don't have that. And so this is actually why we can't take a human gene for insulin and just insert that DNA exactly as it is into bacteria. We have to first let the cell splice it out, and then we can turn that mRNA that has been spliced back into cDNA. We call it complementary DNA. It's complementary to the spliced mRNA. It has the introns removed. It's exons only, and we can turn it back into cDNA. And this is why that exercise that we worked on talking about HIV and what could we do with reverse transcriptase, because this is where reverse transcriptase comes in. It's why you can get insulin affordably now. Insulin used to be a luxury item, and before we were able to extract it from cows, you just died from diabetes, quite literally. You just died from it. And so now we have the availability of insulin, human growth factor, and all sorts of things. Humira, all sorts of things are made because of this beautiful technology that we have to give thanks regarding for reverse transcriptase. A horrible disease, HIV, but it definitely came with some blessings. So we frame sometimes things that way. But anyways, that's the idea of the eukaryotic genes and how they're regulated with introns and exons. We splice out that stuff. I wish there was a way to just get rid of all the drawings. I'm sure there is, and I'm just not seeing it. But whatever. Okay. It talked a little bit about translation, which is whenever the ribosome is going to be reading our codons. So each codon, which is 3 nucleotides on our mRNA, we call it a triplet if it's in the DNA. But if it's in the mRNA, which is what the actual ribosome is reading, then it's called a codon. And again, I talked about this a little bit more in class. This is just my summary. But basically, if your mRNA had AUG, then what'll happen is inside of the ribosome, this is a ribosome, it clamps down on your mRNA, and there'll be these pockets inside. There's 3 pockets, but there's these pockets inside of there where the tRNA will come in. And the tRNA looks kind of like this guy. So it'll have a match to it. So if it's A on mRNA, on the tRNA, it will have U, A, C. Again, because U binds with A binds with U, just like T in the DNA. All right. And this here is called a codon in the mRNA. So in the tRNA, that sequence is called an anticodon. And when those match, that's how the tRNA knows what to bring attached to it. If it's a tRNA that has the UAC in the anticodon, then it will carry methionine and so on and so forth. And we have that whole table of all of our different amino acids that we could refer to that I'm not making you memorize. But that's what your body, basically how it reads your genetic code. It looks at the codon, it matches it up with an anticodon, and based on what your anticodon is, it knows which amino acid to bring in. It'll bring in the next one and link it on with a peptide bond. In the next one, peptide bond, and the next one, peptide bond, and so on and so forth until we have built the whole protein out of amino acids according to those codons. And that is translation. All right. Then we had the operons. And don't you love it? We talked about inducible and repressible operons. In the textbook, it gives example of the lac operon as the inducible operon and the arginine operon as the repressible one. I don't really care that much that you remember, that the lac operon is an inducible one. I mean, it's in your learning objectives. Yes, okay. That's probably the most famous one. So we do need to, so I'm kind of lying to you a little bit. You do need to know the lactose operon, the lac operon. But just understand that there's a lot of different inducible operons and a lot of repressible operons. In fact, in lab 10 next week on Monday or Tuesday, we're going to go over transformation, and transformation not in eukaryotic cells, but in bacterial cells as a means of getting new genes. And how we're going to regulate that genes is through an inducible operon. So we're not going to stop with just introducing this concept. It's going to be one that comes up again. So inducible operon and repressible operons. All operons, and again, I know I talked about this in the thing, but we can't review it enough because everybody is terrible with operons, okay? All operons, we have our promoter. This is in the DNA. My D always ends up janky. In our DNA, we have a promoter region, an operator region, and the genes that are associated with a similar function altogether, okay? In that promoter, we already talked about what the promoter is, and hopefully you'll remember what it is, but this is where RNA polymerase binds, and then we'll start copying to make the RNA by looking at the DNA. Okay, so RNA polymerase binds onto the DNA, opens up the DNA, and then makes a working copy of the DNA. That was transcription. We remember transcription. All right, and then we have an operator region, and then we have genes. So this is all operons. They all work this way. All right, so in a normal situation, get my colors ready, just bear with me. In a normal situation, RNA polymerase will come here and bind on here, and then it will be able to basically move on down and transcribe. It'll open up the DNA and copy the DNA, but making RNA with RNA nucleotides. That's normal situation. This would be turned on. This is an operon that is turned on. I'm going to redraw the same thing. Here's our genes. Here's our operator. Here's where our RNA polymerase binds. Cool. RNA polymerase can bind here. We're starting off the same. But in the case where we are turned off, hold on, something else binds here. And this thing is called a repressor. And you can imagine that quite like you would think it looks like, you would be right. We can't move if that repressor is blocking the way. All operons, in this situation, we're off. This is how we turn off the gene expression because of this. All operons work that way. This is where now, if you can get this down, that this is the two ways that an operon can be. Either on, it's open and it's on, or the repressor is bound and it's blocking RNA polymerase, and that's turning off expression of the genes downstream. You're either on and open, or you're locked and off. And you're locked because you're blocked by a repressor. So that's your two options. If you are that's it, okay? That really is it. If you are an inducible operon, if a woman is pregnant and you have to induce labor, was she already in labor? No. This is where I always start because most people understand that concept. If you are inducible, then you are starting off. Quite literally, you start off and we'll remove the repressor to turn you on. If you are a repressible operon, the only way we can repress you is because you are on. So the repressible operons start on. We will add a repressor to turn it off. And if you want to see me go into more detail with the examples and everything like that, again, I'm going to refer you back to either the lecture or the recording of the in-class explanation. Okay. So that's my watered down version of inducible and repressible operons. They either start on or you start off. And then you turn on or you turn off, you know, depending on where you started, okay? And that's it. That is literally it. Don't overthink it past that. And that's it, okay? Don't try to overthink it past that. All right. We learn a little bit in chapter 6 and 8 about bacterial horizontal gene transfer. This refers to conjugation, which is shuttling DNA copies of usually plasmids through the sex pilus. The pilus is for a channel that connects two cells together. They literally are like a cell is hanging out and a cell is hanging out, and they build a little channel between each other, and then they copy their little DNA and then shuttle it through the channel. So now the other guy can have a copy too. That's conjugation. You guys know what conjugal visits are at a prison. Maybe you've heard of that. Conjugal visits is when the prisoners get to have sex with their significant others. Well, it's the same thing. Conjugation, referring to sex, swapping DNA here, okay? Except it's this time through the sex pilus. This is as close as bacteria will ever get to sex. All right. The other one is transformation. And I understand we've heard the term transformation in regard to our oncoviruses, our cancer-causing viruses, right? This is a different kind of transformation. In bacteria, transformation means that we are changing our DNA. So that's still the same. We are changing the DNA of the bacteria. But in this case, it's because our bacteria, competent cells of bacteria, are picking up external DNA. So quite literally, and we will go over this in lab 10, and you'll get to kind of walk through it, okay? But in transformation, here's some DNA hanging out from, I don't know, a cell got killed, and it's in parts now, and there's some DNA hanging out, and along comes another cell, and it can just take that DNA inside of it and incorporate it into its own DNA. That's transformation. Not all bacteria can do that. Only competent cells can do that. And we'll walk through it in lab 10 next week. It's probably one of the most widely used biochemical things that we do as scientists to manipulate our cells. And then the third one, because there's three different versions, there's conjugation, there's transformation, and then there is transduction. Transduction, we have actually talked about before. This is when bacteriophages incorporate their genes into our bacteria and give them new genes. That's a process we know as lysogenic conversion. And then that's what's happened to the cells. The cells have gone through lysogenic conversion, but the process to cause lysogenic conversion is called transduction. Okay. All right. And then we talked about the different types of mutations. Let me see. Let me clear up my screen a little bit. I need a much bigger eraser. Oh no, that's not going to work for me. So I just have to be patient with this. Great. Erase all this. Get rid of our operons, even though they were beautiful. Okay. All right, mutations. See if I can do this thing. Keep it all right better. All right, there's several different types of mutations. A missense mutation. Nope, I don't like that. I don't like that it made it a tiny box for some reason. What is this? Bear with me for a second. Missense. Nonsense. Silent. If our original was, I don't know, AAA, and it coded for phenylalanine. This was our code, our codon, and this is our amino acid that it codes for, okay? If our original was AAA, our missense might be AGA. It's a point mutation here, right? Whenever you have a change that causes it to, let's say AGA codes, I don't know what it codes for. Let's say it codes for isoleucine. That's a different amino acid. That's a missense mutation. In a nonsense mutation, let's say we went from AAA and we got changed to UAA, and that's a stop codon. It doesn't code for an amino acid. That's nonsense. So that's a nonsense mutation. It causes a stop. A silent mutation. If you went from AAA to AAU, and it still codes for phenylalanine, right? That's a silent mutation. We're still coding for the same amino acid. So it's just whether your change is a different amino acid, no amino acid causing a stop, or the same amino acid. So those are the three basic ones, all right? If you made a change and you went to AGA, let's say that's what you changed to, and now you're going back to AAA, that's a back mutation. Going back to the original. And then if you coded for, let's say, AAA, CCC, GGG, all right? And this is your original. And then you changed to where we remove the A. We take it out. Well, the body doesn't read it as AA space CCC. It moves the whole thing over to compensate. It still goes in threes. It'll be AAC, CCG, and then GG, whatever comes next. And then you could see very quickly how that can become a problem. This is called a frame shift mutation. So obviously, frame shift mutations and nonsense mutations are the worst. You may be fine replacing with an amino acid with another one. Your protein might still function properly. It might not. And any of these changes that cause a drastic change in our protein, leading to a different phenotype. So let's say we change one nucleotide. That changes one amino acid, and that causes the protein to fold differently. That might just lead to different eye color. Or it could lead to a non-functional protein that would lead to a non-viable fetus and be deadly. Or it can cause a dysfunction in that protein, and that could lead to a disease. All of those different versions of the genes that are associated with just one nucleotide, we call those single nucleotide polymorphisms. So single nucleotide is associated with it, just one nucleotide. And we have different phenotypes associated with it that can be good or bad. Polymorphism means many versions. So single nucleotide changes causing many versions of the outcome. That's more of a chapter 8 thing, but I feel like it relates to this. All right. That's my summary of chapter 6. Does anybody have any specific questions about chapter 6 before I move on? All right, I'm going to go into chapter 8. Trying. Okay, here we go. All right, we started off with restriction endonucleases. These guys cut at specific sites of the DNA called palindromes. They cut at the DNA. They cut the DNA itself at palindrome sequences. So what's a palindrome? For example, you might read CAA, TTG. On one strand of your DNA, remember your DNA orientation. One side's 5' to 3'. And the other side is 3' to 5'. Because we're anti-parallel, this is your DNA, and this is your two strands of your DNA. So if you have CAA, TTG, you might have GTT, AAC, because of how these guys match up. A to T, C to G, right? So I want you to notice something. If you read from this to this, we read it CAA, TTG. But if you read this 5' to 3', which would be in this direction, what does it read? CAA, TTG. You see how it's the same, forward and backward? That is a palindrome. You'll also notice that if you just take one strand, the CAA, TTG, if you fold it in half, the two sides match too. This is part of the palindrome. Okay? So that's a palindrome sequence. And then the way that these restriction enzymes often cut will be like right here. And what will lead to, see if I can draw it. You would have a C still bound with a G, and you'll have a T, T, A, sticking out. And this is the rest of your DNA attached to it. And on the other side, you would have a G attached to the C still with DNA. They still got attached to the DNA. T, T, A, A. But they're not attached to each other anymore. We broke that. We've got sticky ends. This is a free end here, and we've got a free end here. That's our sticky ends. And they would want to bind back up with each other. You can see how they would want to. But now let's imagine that we took from our previous slide. Let's imagine for a second that we took, oh, I erased it. That's fine. That's fine. We took our cDNA that we made from our mRNA for insulin, okay? We have a spliced up mRNA. We removed our introns. It's only exons. And we turned that mRNA into DNA that has no introns in it. We called that our cDNA. Now we can put our cDNA sequence and slap on some ends that'll match up with these ends and now insert it into our bacteria genes. That is what we did. Not me or you, but that's what scientists did. That's how we have insulin today. You can imagine that if I already told you about transformation and conjugation and transduction, now you can use those as tools to insert whatever DNA you want to into a virus that infects bacteria and then make the bacteria express that gene into a little plasmid and put your gene into that plasmid using this technology and shove that plasmid into a competent cell through transformation. And then you can actually force those guys to conjugate up with other bacteria, make a copy of that, and then put it into a new guy next to it through conjugation. That horizontal gene transfer just became useful as well. And that is actually how that will work, not the conjugation part, but the transformation. We would take a little tiny plasmid, put insulin into it, the human insulin cDNA, and then mix it up with a whole bunch of bacteria, heat the bacteria up to open their pores, mix it with so much plasmid that it's going to be hard to miss the plasmid. And eventually some of these guys will take up some plasmids. And not only can we engineer that plasmid to have insulin in it, but we can engineer that plasmid so that insulin is operated and triggered by an operon, which is literally what we're going to talk about in Lab 10. Okay, so we're using this as a mechanism to force bacteria to produce human gene products in excess so that we can deliver them as insulin to our patients or human growth factor to people that need it, or even other biologics as well. And this is so widely used, you don't even know, man. All right, so anytime that we create something like this, where we have forced bacteria, for example, like maybe this one, to carry new genes for us, this bacteria is now a transgenic organism. It's got genes from another organism in it. Another word for transgenic is GMO, genetically modified organism. And so we can do this to bacteria, and obviously we can do this to other organisms as well. We can do this by crossbreeding them or by genetically manipulating their DNA. So we have quite a lot of things out there in our environment now that have been genetically manipulated this way. We try to enhance organisms this way. One thing I want to make very clear that I talk about in the lecture, but I can't drive it home enough. If you ate this bacteria here, why would you think that you would get this gene incorporated into your DNA? Don't we only wish that for our people that have diabetes? I wish it was that easy for you. You could just eat this bacteria and this DNA would get incorporated in your DNA because that's apparently what people think transgenic organisms can do to us. That is wild that you think that. Not you, but we know who we're talking about, right? That is impossible. Okay. That's not the risk with transgenic organisms or GMOs. That DNA that we engineered into that organism, whether it is a bacteria, a chicken, corn, whatever. And literally every single piece of corn you've ever had in your life was a GMO, by the way. I can almost assure you. Okay. But those genes can't jump out of that organism into your genes. That is literally impossible for that to happen. Okay. Period. What this is a problem with is that we are inserting genes like pest resistance. So there are genes that we took from bacteria that we inserted into corn to make it so that the corn was resistant to insects. And as a result of doing that, that corn is now resistant to insects. That's great for farmers. It's not great for the environment. So that's the real problem. Plants can transmit their genes to other plants, not to you, but to plants. So that gene that we put into corn, that gene, if we put it into corn, it could transmit that anti-insect gene to weeds. You see how that's a problem now? It's a big problem. And it has been shown that it does happen. That's our bigger issue. How does this affect the ecosystem on a larger scale? But the issue is not that it can jump to you. That is not the issue. So please don't think that that is it. You're eating GMOs. You've been done eating GMOs your whole life, and you ain't turned into a corn or a bacteria yet, and you're not going to. And I kind of wish that when we put the insulin gene into the bacteria, that it could jump into the people that have diabetes. I'm sure they wish that too, but that's not the way that life works. As it turns out, if this was a normal bacteria, just a normal bacteria without the plasmid, without transgenic, and it has its own DNA, not any modified DNA. So this would be our GMO guy, and this would be our normal bacteria, not GMO. When you eat this guy, and you do, if this is bacteria or corn or chicken or anything, any plant or animal or bacteria or yeast, they have DNA, right? When you eat this DNA, their normal DNA, their non-GMO DNA, if you have organic non-GMO food, it still has DNA. Why doesn't that DNA come incorporated into your genes? Why would you think that DNA? Why aren't you worried about that DNA? But you're worried about this one. It's still just DNA. It's still literally just DNA. You eat DNA every day, whether you're eating plants or animals. They both have DNA. You eat DNA every day, or you're dead. Like, that's it. Where do you think you get your nucleotides from to build your new DNA? You can eat DNA and steal your food's DNA and turn it into the building blocks for your own DNA. You can also break that DNA down so far that it's not even DNA or nucleotides anymore and use it for the parts for other things. But you eat DNA already. Just saying. Maybe you didn't think about it that way. All right, no, we're not done yet. I'm trying to erase this and I clicked the wrong button. Okay, so just want to say that. Just want to clear that up. You eat DNA anyway. Why is GMO DNA different? I am not an advocate for GMOs. I want to be clear about that because I think they harm the environment, but they're just harmful for different reasons than what people typically think. Okay. All right, the three, I guess there's a few things I want to talk about with our tools. Nucleic acid hybridization. Nucleic acid hybridization means that, let's say, cancer cells have a sequence in them, I don't know, AAA. And we know this because we've studied it as molecular biologists, okay? So you can make a probe that says TTT. This is your probe. It's some DNA. Obviously, it's a way longer sequence than this, okay? But it's just me saying this. If cancers have AAA sequence, and you can take your biopsy from your patient, from their breast cancer, and mix it with the TTT and link it to a fluorescent probe that glows. You can literally just take your patient's biopsy, match this probe to it, and see if your patient's cells glow or not. If they glow, they have cancer. That is nucleic acid hybridization. If the sequence of the DNA matches up to your patient's sequence, then your patient has that target sequence. We can look for cancer in the presence of specific infections this way. So if we took a swab from your patient's throat, we could put it and mix it with a probe for Staphylococcus aureus and see if it glows. And if it does, that person has Staphylococcus aureus. So that is nucleic acid hybridization. All right, another tool, PCR. PCR is way easier than you ever thought it was. In PCR, PCR stands for, and you do need to know what it stands for, and you need to be able to recall it and spell it on a test. Polymerase chain reaction. It's right here. All right, polymerase chain reaction. There's only 3 steps. First step is denature. In the denature step, we're taking our double-stranded DNA and splitting it apart to single strand. We use heat to do that. And in the second step, we anneal. We will take our single-stranded DNAs and we'll get a primer that matches up. All right, that's the anneal step. In the elongate step, let's see if I can do this. I just wanted it as like one thing, but sure. You don't even know. Like on my end, this looks really crazy. It's not letting me copy it, but that's okay. Whatever. So in the elongate, I'm going to draw it just in a different color so you can see that it's a different thing. Elongate. Okay. We're going to build off of it. Match, 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 match. And then build off of this one. Match, 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 match in one direction. Okay. That's the third step. That's it. So now we went from 1 strand and we made 2 out of it. And it works exactly the same as replication. Look at that. Our blue strand is our old strand and our red and green is our new strand. So it is semi-conservative, just like replication is that we learned about in chapter 6. And so that's our first cycle. We would go from 1 to 2. In our second cycle, we would have started with 2 strands and 2 become 4 because quite literally every cycle you go from 1 and it becomes 2. You go from 2, each one of those 2 becomes 2. Each one of those becomes 2. So 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32, 32 becomes 64, and so on and so forth. So this is after 1 cycle, after 2 cycles, after 3, after 4, after 5, after 6. And you better bet I will ask you, if you start with this number, how many will you end up with after this number of cycles? That's just doubling. That is not hard math, right? So I warned you there would be easy maths, and that's one of the easy maths. If you don't understand why this is an easy maths, I don't know how you got this far, okay? Because you're going to fail at dosage calculations. So you need to work on that, all right? If that's overwhelming for you. All right, so that's PCR. You will notice, I should have never erased it. You will notice in PCR, let me erase this stuff so I have a little bit of room to work with. In PCR, I'm going to leave my major steps of PCR up here. But in PCR, let this catch up so you can see. I'm actively doing it on my screen, but you guys don't see it until I click off of it. We got the separate strains. I want to keep that up there. You can see how in PCR, we rely pretty heavily on those primers. Those primers have to match the sequence. The primers determine what you're making the copy of, okay? So you can't just have, hey, here's somebody's swab of their nose. What do they have? Run it in PCR and find out. That's not how it works. You get a swab of their nose, put it in PCR with primers for a specific gene. And that gene has to be specific to an organism. Otherwise, you're not getting specific results. So these primers are usually about 30 nucleotides long. And we're talking about genes that are thousands of base pairs long. And so the idea here is basically that that specificity is like if you make a copy of DNA, if you can elongate, your primer's bound. If your primer's bound, that's because you were positive for that thing. It is that simple. That's when I say PCR is simple. It quite literally is. If you elongated, your primer's bound. If your primer's bound, they bound to your target sequence. Your target sequence was there to bind to. So if you're checking for COVID, your primers bind to a COVID sequence. If you're checking for strep throat, your primer's bound to a strep throat sequence. And it's all been designed so it's very specific for those things. If you elongated, that meant your primer's bound to a COVID sequence, and therefore you have COVID. Yes, what's your question?
So is this where they got like the molecular testing like in clinics and stuff? Is that what they use?
This is what they use. They use PCR. So
I
it's
see.
exactly like this. And so they have to have the primers for that thing. And so almost always, most clinics, even urgent cares, they'll have primers for our most commonly tested for things. But other tests, they'll have to mail it out to somebody else that has more primers for other diseases, right? So it's pretty common to have PCR at like urgent cares or basic like general doctors clinics, have PCR that can test for flu, COVID, and RSV. But for example, they're not going to have PCR that tests for the plague, right? And so if they want testing for PCR for the plague, they'll have to mail it to somebody that has the primers and the PCR set up and program for the plague specifically. So yeah, so that's that basically. And that's how it works. If you elongate the sequence. And so how do we know if it's elongating? Well, the PCR works the same way as everything else. If you have a sequence A, T, T, C, G, then as we're adding on we separated our strands, right? If as we're adding on nucleotides, we'll add on a T, and then we'll add on an A, and we'll add on an A. And in PCR, what we do is we'll label the T with a color. We'll label A with a color. So I'm using one color here, but let's say A is red. And I don't know, C is blue. And whatever. So when a G binds, this would be G, right? And the other one will be C. This is not a good color. And C is purple, let's say. Okay. So when a T binds, we'll get green flash. When an A binds, we'll get red flash. When a G binds, we'll get blue flash. And when a C binds, we'll get purple flash or whatever. And the machine can detect these colors with lasers. And as we're adding the different colors, it's verifying that, yes, these colors are coming in the correct order, and these are actually flashing the color they're supposed to. And so we can get those happening in real time from a detector while this is happening. And so PCR over the course of an hour, we are just separating strands and trying to elongate. And if that is repeatable over a period of time, then we say that that person is positive. It can happen as a fluke. So that's why we want to do it repeatedly over time and make sure that it is a real result. And that's why it takes an hour, because we're just going from hot, adding on our primer with a slight cooling, and then elongating, and then going hot again, separating. Add on a primer, and then elongate, and then heat that up and separate, and then add a primer, and then elongate. And the machine does all of that for us, but that's the idea. And then we're looking for color flashes. to see if we're adding on certain nucleotides. And that's how PCR really works. All right, any other questions? I feel like PCR is so commonly used, but not understood how it works. And that's it. This is it. All right, so the other one that it talks about is gel electrophoresis, which is a little bit easier. When we talk about our DNA strand being a sugar phosphate backbone, sugar phosphate, sugar phosphate, sugar phosphate, don't forget that what that phosphate really is PO4. And it looks a lot like this, P with an O and each of these O's on it. And all of these O's are negatively charged. So DNA, because of all of these phosphates that we have over and over again, DNA is very negatively charged. All right, so we're starting with that. Let me see what I want to erase. Let's erase this. We're starting with our negatively charged DNA. Go back to the purple. And if DNA has a negative charge to it, and we're going to put it inside of this gel, and let's say there's some wells in the gel. You know how these look, okay? They're literally like gel, and they're agarose, which is the sugar made from agar, okay? So it's a lot like the agar that we work with on our plates, but it's just purified. And then what we do is we run electricity through it. And because DNA has a negative charge, it's going to run to the positive end. Opposites attract. And so we put some DNA in the well, run electricity through it. And just like if I had a big old mesh screen that you had to walk through, the bigger you are, you're going to have a harder time pushing through the screen. But a little insect could crawl through the screen or fly through it really easily. So bigger molecules get stuck in the gel, and smaller ones travel faster, and they create these bands. So the further towards the bottom you are, the smaller you are. It says smaller, I wrote off the screen, I apologize. So small towards the bottom, the large towards the top. They get trapped in the gel. Just like if there was a crowd of people, smaller people could make their way through the crowd easier. So the big ones get trapped at the top, and then the small ones travel faster. And this is also where we come back to that restriction endonucleases. If I cut my DNA randomly with one type of restriction enzyme, then my gel might look like this, but yours might look like this. And so this is how we can identify comparisons between people's DNA without having to sequence their entire DNA. Just look at how it behaves when we cut it with restriction endonucleases. So that can be useful for that. It's also used to purify DNA sequences and things like that for use as tools. For example, if we had that plasmid from earlier that we inserted the insulin gene in, but now you want to work with just the insulin gene, we can cut it with that same enzyme, run this on the gel, and then separate out the plasmid from the insulin gene on the gel, and then purify out just the gene and work with just that, or whatever we wanted to do with it. And that's how we manipulate our DNA. Smaller fragments run faster. What else do I want to say about this? We also talked about gene therapy a little bit. We talked about this more in our lecture. We can often use viral latency. So we know about lysogeny, and that can happen in people and in bacteria, right? That the virus inserts its genome into the host genome, and that leads to, in eukaryotes, we call that viral latency. And HIV does that, but we can hijack that and replace, again, using a lot of the same tools. If this was an HIV genome, cut out the HIV stuff and put in a gene for something else. And let's say it's a gene that codes for a form of hemoglobin that works the way it's supposed to, as opposed to the messed up version of hemoglobin that led to sickle cell trait. And that's what the example was in our activity. Our activity talked about sickle cell disease. And we can replace that trait with a working version of the gene by inserting it into HIV and then forcing HIV to insert that instead of HIV BS. So now we have changed the genetics of the patient. So now it is expressing hemoglobin the way that they're supposed to. And you might think now they also have HIV. No, they don't. We removed the HIV stuff from it. and just used the shell of the virus as like a missile and carried the good genes with it. But HIV would only infect T cells, for example. Now, we can engineer our viruses to infect whatever we want them to infect if we understand the virus well enough. We don't want to create a virus that causes disease that does that. But let me tell you, scientists can do that. How do you think COVID came to be? They invented a virus that basically was SARS and then engineered it to be more infectious, right? I mean, that's what we think, anyways. So this is the same idea. We're not trying to do that here, though. We're trying to say, let's remove the bad stuff, insert a good gene, and use this for good. Then we can make this virus infect whatever cell group that we want it to and insert whatever good gene that we want it to. Going through the clinical trials and things like that for these things is incredibly expensive. But there are, I think, 13 different versions of gene therapy that are functional nowadays. Just 13, but it's a start, and they do work often very similarly to this. But using viruses as a means to get a new working version of the gene into those patients. This only affects right now somatic cells. This is non-heritable. It's like if you change the DNA of your muscles, will that DNA get passed on to your kids? No, your eggs and your sperm don't come from your muscles. So that's the example here. We're changing the DNA in your, typically it's in your bone marrow. Those are stem cells, but not all of your body cells come from your bone marrow, right? Your blood cells do. And your gametes, your sperm and your egg, don't come from your bone marrow. So changing the bone marrow doesn't make it heritable. But it does change that organism's genetics in their body permanently, just not spread to their kids. So if we want a genetic change to be passed on from generation to generation, then those genes have to be changed in the gametes or the early embryo. So that could be done at the IVF stage of things. And then that would be heritable. All right, any questions about this stuff? So I'm gonna move on. We're gonna knock out our questions.
Professor Yost,
Yes, what's up?
I was curious, for the PCR steps, it could step 2 be called priming?
Yes. Yes.
Okay, I just want to make sure I didn't, because that's what I put down and I was going back like, oh, how did I miss that? So,
Yeah, it can be called priming. Just be aware that it can also be called annealing. So
okay,
just a matter of, I guess, like vocabulary stuff. What I mean is annealing means for things to stick together. When you anneal something, it just means you're sticking them together. So it's the same thing. So just be aware of
okay,
that word. And if you guys get on a test and you're like, you want to call me over, right? And you get to a question on a test and I put anneal, and you're like, so I know that what happens is the primers bind to the complementary sequence, but I can't remember that anneal is the word for that. You can call me over and I'm not going to cry about it. Okay. So FYI, if you have that on a test, feel free to call me over and tell me that you know the concept and I'll help you out with the term if that ends up being an issue.
no, I've already changed my notes. It's annealing now.
Anneal,
Thank
yes. Yes. All right. Any other questions about this? All right, awesome. So we'll move on to our most missed topics for this week. I'm trying to click on this and it is. Wow, that really went far ahead. Okay. Okay, we're here now. All right, for chapter 6, bacterial transcription versus eukaryotic transcription. They are different. So for the bacteria versus the eukaryote, the first things that we want to start with, back in unit 1, what did we say the difference is between bacteria and eukaryotes? Don't worry, I'm getting there. Bacteria have no nucleus and no organelles, no membrane-bound organelles. That's a G. Eukaryotes do have a nucleus and membrane-bound organelles. All right. So how does this change transcription? In bacteria, you just got a cell and you got some DNA in there. So we can take the DNA, make mRNA and protein, turn it into protein. That's all happening in the cytoplasm. There is no nucleus. So what bacteria can do is something called co-transcriptional translation. I'll write it on here. It's in the lecture, but it is called co-transcriptional translation. What this means is, because there's no nucleus in eukaryotes, when you have a cell, you got your DNA in the nucleus. If you want to work with your DNA and make RNA, that has to happen in the nucleus. So transcription has to happen in the nucleus. Then you take the RNA out and you can translate it. You don't got to do that in bacteria. So in bacteria, because they're all in one place and it's all happening in one place, while you are making your RNA, so let's imagine this is your DNA in bacteria. You've opened up your DNA. You're using one side of your DNA to make a copy to make RNA. Whatever, there's your mRNA. And after you have done that, your ribosome, while you're doing that, your ribosome can clamp on and start matching these guys up on your tRNA and attaching the amino acids to make protein. And that can happen all at the same place at the same time in bacteria. That is co-transcriptional translation. While we are transcribing, we can start translating. We don't have to finish the mRNA. Co-transcriptional. While transcription is happening, that's co-transcriptional. And then translation happens while we are transcribing. It's wild. But bacteria can do that, and eukaryotes can't, right? The other thing, so I'm going to erase this picture because I don't want taking up everything here. All right. The other thing eukaryotes can do is that splicing. I say they can do, whether you think it's a good thing or bad thing. That splicing of introns and exons. I need more space. Bacteria don't have to do that. Those are the general things. Oh, and operons. Bacteria have operons. Eukaryotes don't have that. Those are the major differences, okay? And this slide summarizes that a little more if you need more info past that. But I'm going to move on to the questions because I don't have a lot of time. I have lab at 6:45, so I don't want to take too much longer on this. Okay, so which of the following statements accurately differentiates bacterial transcription from eukaryotic transcription? Bacterial versus eukaryotic. So we could go back to that previous slide where we made some notes about that stuff. There's our little, I want to say cheat sheet. We'll come back to it, okay, if we need it. Bacterial transcription lacks RNA polymerases. No, we're still making RNA using RNA polymerases. So that's not it. Eukaryotic transcription occurs in the cytoplasm. No, eukaryotes, we had the cell, we had the nucleus, we had DNA in the nucleus. And in order to go from DNA to RNA, that's transcription, right? In order to make RNA, that has to happen in the nucleus, not the cytoplasm. So that's already wrong. Eukaryotic transcription involves extensive mRNA processing. Ooh, yes, that was our splicing. While bacterial transcription does not. This one seems like a good choice. Bacterial transcription uses introns and exons. So no, that is eukaryotes. So it's not this one. So C was our answer here. A new drug is being tested for its ability to inhibit transcription in bacterial cells. So bacterial cells. Which of the following mechanisms we're trying to inhibit it? So inhibiting DNA to RNA in bacteria. So this doesn't need a nucleus if it's in bacteria. That's our key there, right? Which of the following mechanisms is the drug most likely targeting to achieve this selective inhibition. So only affecting transcription in bacterial cells and not affecting us, right? Is it affecting the DNA polymerase in bacterial cells? So I want to point this out. Does transcription going from DNA to RNA, does that need DNA polymerase? It doesn't. That is replication, not transcription. B, the mechanism that shuttles mRNA out of the bacterial nucleus. Y'all don't get caught. Trick. The RNA polymerase activity in the bacteria, maybe. We do need to have RNA polymerase activity, maybe. The splicing of introns from eukaryotic mRNA transcripts. How would that inhibit transcription in bacteria? So let's see. It's going to target the RNA polymerase activity in the bacteria specifically. All right. Questions about that? We're going to move on. All right, next part. Operons. Okay, so this goes into a little bit more detail, talking about those operons. I'm not going to go over this slide. What I am going to do is draw our little cheat operon setup when we answer the questions here, okay? I don't have a ton of space on this. All right, let me see. No, okay, here we go. When in all operons, we have a promoter, which is where RNA polymerase binds, already starting to lose it. An operator, where our repressor binds, and all of our genes. I'm going to draw this twice. We're going to segment it off. Again, this is only in bacteria. And this is in their DNA. This is RNA polymerase, and it will be able to transcribe. I'll just draw it really thick. We'll pass that operon and make the RNA from this. So this is on. In the case of an off version of an operon, RNA polymerase, repressor, it can't go. No genes are made. This is an off operon. Now it's just a matter of where you're starting. So if you are inducible, you start off. You can be turned on. How do we turn on? By removing the repressor. In the case of the lac operon, the way we remove the repressor is by adding an inducer. In the case of the lac operon, the inducer is lactose. That's why it's called the lac operon. So lactose binds to the repressor and causes it to come off. That's how the inducible operons work. And the repressible, they start off. Sorry, they start on. Start on and can turn off. I'm getting ahead of myself. By causing that repressor to bind. To the operator. All right. Okay. Which of the following statements accurately describes a key difference between inducible and repressible operons? Inducible operons are typically off. This is true. and are turned on by an inducer. Okay, I'm tracking. While repressible operons are typically on and are turned off by a repressor. This is true. So it's probably this one. We're going to read through our other options and weed them out. Inducible operons are always on. No, you're not inducible if you're on. You're repressible. That one's wrong because of that. All inducible and repressible operons are regulated by lactose. No. In fact, if I were to say all inducible operons, just take this part out, are regulated by the presence of lactose, that would also be false. Only one version of inducible operons are regulated by lactose. The one we're going to see in lab 10 is regulated by the presence of arabinose, which is a completely different sugar. They're not all regulated by sugars either. All right, next, inducible operons and repressible operons both use the same type of repressor protein for regulation. Not always the same one. The repressor protein for the lac operon responds only to lactose. The repressor protein for the arginine operon responds only to arginine. So if they respond to different things, they're different proteins. So they're not the same proteins. All right. A pharmaceutical company is developing a drug that aims to inhibit the expression of genes involved in antibiotic resistance. Okay, cool. The drug specifically targets the repressor protein that regulates a repressible operon. So something that is on that can be turned off, and this gene regulates that. Which of the following would be a likely outcome of this drug's action on the bacterial cell? Okay, the operon will remain off. Well, it's a repressible operon. It wasn't off and remaining off. It's on. So that's already starting off wrong. The operon will be activated in the absence of the repressor, leading to increased expression of antibiotic resistance genes. Maybe this is the wording is weird on this, so I want to come back to it maybe. The drug will have no effect on the operon since it only targets inducible operons. It literally says repressible. The operon will be turned off regardless of the presence of the inducer, leading to decreased expression of antibiotic resistance genes. This is a repressible operon. There isn't an inducer. Inducers are in inducible operons, so it's not D. It's going to be B on this one. Did I have any notes for this one? I don't even know if I do. I do have it. Let's see if my explanation is clearer than what I'm trying to explain it as. The operon will be activated in the presence of the repressor. So, I mean, this makes sense to me when I read it. I just feel like I can't draw it out clearly with the space that I have here. Let me see if I can. The operon is activated in the absence of the repressor. So what we're looking at here, I hate how much stuff is written here. I hate it. If we start on and we have changed the genes so that you can't get your repressor, which is in red here, then the operon stays on. That's what this is talking about. So the drug prevents the repressor from blocking. So the genes just stay on. So that will lead to the genes for antibiotic resistance staying on, which is the opposite effect of what you would want. So this would not be a good choice for that drug if you're trying to block antibiotic resistance. Okay. I know that was a complicated one, but that was the thought behind it. Luckily, I mean, hopefully to me, my questions on my test aren't this hard. So hopefully if you can understand this one, the test questions should be easier than that. All right, so that's it for the chapter 6 questions. Do you guys have any questions about those questions? Questions about the questions? Okay. Chapter 8. Okay, so this one's going to be starting off with restriction enzymes. These are the guys that cut at the palindromic sequences and create the sticky ends. There's some key characteristics about restriction enzymes. They are very specific. So there is one enzyme that cuts at CAATTG. All right. That has the GTTAAC. That cuts like this. There's one enzyme that cuts like that. There's one enzyme that cuts at a sequence GATATC. Yes. That cuts like this. There's one enzyme that cuts at one place, one enzyme that cuts at another place. They're all at palindromic sequences. Any potential palindromic sequence that you can think of, there is one enzyme that cuts at that one spot. Some of them create sticky ends, like what we're drawing here, but some of them cut straight down the middle and create blunt ends. There's all sorts of different enzymes. These enzymes are made by bacteria as a defense mechanism against foreign DNA. And we've just hijacked this system to cut at these specific palindromic sequences. All right, so we use this for DNA cloning technologies with sequences that we know, and we can engineer these types of sequences into our DNA so we can cut and paste things together. Let's get into our questions about this stuff. Again, I'm going to draw it out here so you can remember what a palindrome is. CAATTG. All right. Which of the following best describes the role? Let me draw my cuts too so you can see that it is sticky ends. All right, which of the following best describes the role of restriction enzymes in molecular biology? Are they themselves used to amplify DNA sequences by replicating them multiple times? Restriction enzymes don't do that. That's what DNA polymerase does in PCR. This is PCR. Do they cut at specific nucleotide sequences, creating fragments with sticky or blunt ends? They do this. Do they synthesize new DNA strands by adding nucleotides to a growing chain? No, that's DNA polymerase. Do they bind to specific RNA sequences to inhibit their translation into proteins? No, that's a thing too. Those are known as microRNAs or like silencing RNAs, siRNAs, miRNAs or siRNAs. So you can imagine if you have an mRNA that has the code AAUU, I can make a microRNA that says UUAA and bind these things two together. So now tRNAs can't stick to them. That's how silencing RNAs work. This is technology too, and we don't talk about it much in our book, but it is a thing, just FYI. Why is this not updating my color? I drew all this stuff on here that you guys can't see. Whatever. All right, the next question, let's move on. The next question. Oh, I didn't, okay, that's why. Here you go, okay, sorry. Like, why are you guys not seeing what I'm seeing? It's because I was on a different slide than you. All right, now you can see what I'm talking about. Okay, next question for real this time. A clinical laboratory is using restriction enzymes to diagnose a genetic disorder. The disorder is caused by a specific mutation that alters a restriction enzyme recognition site. Okay, so what this means is that a normal gene would lead to running it out on a gel. A normal gene would have maybe 3 because that thing cut at a couple of sites. And now we have a mutation at one of those sites. So now instead of having 3, we have one bigger, longer one, and maybe one smaller one, because one of those sites was changed in a sequence. That's what this is talking about. So cut sites, restriction enzymes in a genetic disorder, we have a mutation that alters a restriction enzyme recognition site. That means a cutting spot. One of those palindromic sequences was altered because of a mutation. And now we don't cut in as many places on that person's DNA. How would this mutation affect the results of the restriction fragment length polymorphism? This word, y'all, RFLP, restriction fragment length polymorphism. What that means is if my genes look like this and yours look like this, but we're cutting them with an enzyme that cuts at this spot, those are restriction enzyme, restriction fragment length polymorphisms, RFLPs. The fragments made from cutting with the restriction endonucleases lead to different lengths. That's what a polymorphism is. Polymorph means many versions. And many versions of restriction fragments as a result of cutting with this enzyme. Okay, so the question is, how would this affect their profile? Well, it would remove a cut spot. The mutation would create a new recognition site. No, we've eliminated one of the recognition sites. Mutation would disrupt the restriction enzyme's ability to cut at one site, leading to the absence of the expected fragments. That's exactly what we drew on our little predictor gel. The mutation would have no effect on the enzyme's activity, resulting in the same pattern. No, we've changed one of the cut spots. We're going to have a different pattern. The mutation would enhance the restriction enzyme's cutting efficiency. Nothing about that would change. It's only the number of sites. One of the cut sites was eliminated, so therefore the fragments that we see on the gel, we would have different fragment lengths seen. Yeah, sorry about that. I don't know what happened with that. All right. Questions about this now that we're caught up with the same slide? All right. Make sure that I'm not missing the slides again. Okay. The next one is PCR. These were our 3 steps: denaturing, annealing, extending. In denaturing, we're going from our double-stranded DNA to 2 strands. In annealing, we are taking those 2 strands and priming them, right? Adding our primer. Add our primer. In extension, we are elongating from the primer. Remember that DNA polymerase is the enzyme that is adding on the nucleotides here. It always is. Same as replication, okay? So when we add on new nucleotides, it's always going to be in the 5' to 3' direction. And it's going to be adding on nucleotides according to A matches with T and G matches with C. And so we'll add on one, and then the next one, and then the next one, and then the next one, and the next one, and so on and so forth on each of those strands. And so we have gone from 1 to 2. I hated how I drew that, but you get the idea. It's a little clearer, that lighter color. Okay. And then just repeats over and over and over and over and over again. It does not do it one time. It does it like 30 to 60 times. Okay. So eventually, in one, if we let the PCR machine go for 3 hours, which is typically what we will do, if we let it go for 3 hours, let it do it as many times as possible in that time, we can go from 1 DNA molecule to billions in a matter of hours. And so that's how we actually detect just one viral particle when somebody has like COVID or something. If it's there and we can amplify it, if it does amplify, you are positive, okay? So here's our questions about that, and we'll come back to our image if we need to. To separate, okay, sorry. Which of the following best describes the purpose of the annealing step in PCR? Remember, the anneal step was just like, I think it was Brandon that was saying. Was it Brandon? It was Brandon that said it, right? Talking about the priming.
Yes,
Okay.
that was me.
I heard your voice and it didn't tell me who was talking. Okay. So that was the priming step. I'm assuming it was Brandon. So that was the priming step. So in the annealing step, now let's find that priming step in here. Is it to separate the double-stranded DNA into single strands? No, that's denature. So it's not A. to allow primers to bind to their complementary sequences on the single-stranded DNA. That is exactly it. To synthesize new DNA strands by adding nucleotides, that's elongating. Or is it to amplify the DNA by increasing the temperature? We actually don't amplify the DNA by increasing the temperature. We have to lower it a little bit for the enzyme to do its work. We only increase the temperature to denature, not to elongate. All right, so the next question. A clinical laboratory is using PCR to detect the presence of a specific pathogen in a patient's sample. If the test is successful, what would the outcome be with PCR? If PCR is positive, then that means we should be elongating. If the PCR is negative, we would have no elongation. Period. That's just how it works. So let's see if any of these choices line up with that thinking. The PCR reaction produces no DNA amplification. If it is positive, we should have amplification. Negative would have no amplification. The PCR reaction yields a single large DNA fragment that is larger than expected. This is wrong because what we're doing with PCR is we're taking that sequence. If this is your sequence, 1, 2, 3, 4, 5, then we were copying exactly 1, 2, 3, 4, 5 and making just 2 of them. We're not changing it in any way. We're literally just copying exactly what it is. So it shouldn't be larger than expected. The PCR reaction generates multiple DNA fragments of the expected size. We'll have multiple because we're making copies. That's all PCR is just a copy. It's a molecular Xerox machine, like it says. The copy machine, indicating successful amplification of the target sequence. This is probably our answer. The PCR reaction results in the formation of a new type of primer that binds to unrelated DNA sequences. That's weird. Why would it do that? We have our understanding of how PCR works. We know it is actually this simple. We are literally just copying the DNA. That's it. Nothing weird else about it. Okay, any questions about PCR or any of these questions? Or any last minute questions for me for this review for chapter 6 and 8? Because if you don't have any other questions, I think that'll be it for today. All right, of course, if you guys think of any other questions, you can always email me. On my online class, you guys have a discussion forum that you guys can post your questions in. I posted a thing that a student emailed me. You're always welcome to use that at any time. Any of my in-class people, if you guys email me with any questions like that, then I can start a similar forum like that for you guys. It's just I interact less with my hybrid class in person, so they have less opportunity for that. But if you guys want me to start something like that, I can. We do have the Unit 2 discussion board that's open as well for you guys to start discussions in there. But you're welcome to bring that up either in a discussion board or email me, and I can answer it for you. So happy studying. Get chapter 6 and 8 under your belt so that next week we can come ready to do chapters 9, 25, and 10. If you don't have any other questions, then I think that'll be it for me today.
Thank you, Professor Yost.
Yep, thanks guys. I'll see you next week.